Hello friends, welcome to some biology class. Friends, today we shall discuss about glycogenesis, the process of synthesis of glycogen from glucose 1-phosphate is known as glycogenesis. It occurs mainly in the liver and in the muscle. Various enzymes that participate in this process are pyridine diphosphate glucose pyrophosphorylase, glycogenin, glycogen synthase, and amylo 1416 transglycosylase, which is also known as branching enzyme. Now let us discuss about the individual reaction steps. As in glycolysis, glucose reacts with ATP to form glucose 6-phosphate and ADP. This reaction is catalyzed by hexokinase in muscle and by glucokinase in liver. In this reaction, ATP is the phosphate group donor. After that, glucose 6-phosphate is isomerized to glucose 1-phosphate by phosphoglucomutase. In the third reaction step, glucose 1-phosphate reacts with uridine triphosphate to form uridine diphosphate glucose pyrophosphate. This reaction is catalyzed by the enzyme UTP glucose pyrophosphorylase. This reaction proceeds in the direction of formation of uridine diphosphate glucose because the enzyme pyrophosphatase hydrolyzes pyrophosphate to 2-phosphate, thereby removing one of the reaction products. So removal of pyrophosphate favors the formation of uridine diphosphate glucose. Friends, glycogen synthase is an enzyme that can only extend an already existing alpha-1,4 linked glucan chain. This enzyme cannot simply link together two glucose molecules. It can only add glucose molecules to the C4-hydroxyl group, which is present on the non-reducing end of a alpha-1,4 linked glucan chain. Then how glycogenesis is initiated? The answer is glycogenin. Glycogenin is a protein. It is a homodimer of 30 kilodalton subunits. This protein acts as glucosyl transferase. In human, there are two isoforms of glycogenin. One is glycogenin 1. It is encoded by GYG1 and is expressed in muscle. The second isoform is glycogenin 2, which is encoded by GYG2 and is expressed in liver and cardiac muscle, but not in skeletal muscle. Glycogenin acts as a primer by polymerizing first few glucose molecules. After that only other enzymes take over. First, glycogenin autocatalytically transfer a glucosyl unit from uridine diphosphate glucose to the hydroxyl group of tyrosine 194. After that, this glucan chain is extended by glycogenin itself up to 7 additional glucose units. So this is the glycogen primer, which is synthesized by glycogenin. And in this primer, the glucose molecules are linked by alpha-1,4 glycosidic bond. So after the formation of glycogen primer, the enzyme glycogen synthase extend this alpha-1,4 linked glucan chain by transferring glucosyl unit from uridine diphosphate glucose to the C4-hydroxyl group, which is present on the non-reducing end. So the enzyme glycogen synthase is responsible for chain elongation, and this enzyme exists in two enzymatically interconvertible forms. One is the original dephosphorylated form, which is more active, and the other is phosphorylated form, which is less active. In human, there are two isoforms of glycogen synthase. The human muscle glycogen synthase is a homotetramer, and each subunit has 737 amino acid residue. And the other isoform, which is expressed in liver, is also a homotetramer in which each subunit has 700 amino acid residue. So the enzyme glycogen synthase is responsible for extending the alpha-1,4 linked glucan chain by generating alpha-1,4 glycoside linkages. Next step is branching. Branching is accomplished by another enzyme known as amylo-1,4,1,6 transglycosylase, which is also known as branching enzyme. Branching enzyme catalyzes the transfer of a 7 residue segment from the end of one chain to the C6 hydroxyl group of a glucose molecule that is present on the same chain or another chain. And the transfer segment must come from a chain which has at least 11 residues. And the newly formed branch point must be 4 residues away from other branch points. So the formation of branch point is due to the generation of alpha-1,6 glycosidic linkage. The enzyme amylo-1,4,1,6 transglycosylase, it generates alpha-1,6 glycosidic bond. So friends, this is the newly formed branch point, and the glycosidic bond formed here is alpha-1,6 glycosidic bond. So the enzyme glycogen synthase extends the glucan chain, and at the same time the branching enzyme establishes several branch points. So by the combining activity of glycogen synthase and that of the branching enzyme, at last a highly branched glycogen molecule is generated. So friends, this is all about glycogen synthesis. For more updates, please subscribe Sam Biology class, and also please share your comments. Thank you very much. Bye bye.